Okay, welcome to Chapter 2, Mitosis and Meiosis. This chapter we're going to cover how DNA is divided into daughter cells after cell division, or in the process of cell division. And there's two main processes that we're going to discuss, mitosis and meiosis. So both what happens and when each of those are used. So by the end of this chapter, you should be able to differentiate between a genome, DNA molecule, chromosome, homologous chromosome, and sister chromatid. So just in general in this genetics class and in general in all biology classes, it's very important to understand scientific jargon that is commonly used. So at the beginning of all the chapters, I'll try to give you examples of important ones for that chapter. So typically, you might have some sort of idea in your head of what a genome is, what a chromosome is. You might think you know exactly what it means, but it's important just to remind yourself that these have very detailed definitions and to kind of revisit these words, especially after you might be reviewing the chapter, just to make sure you can distinguish between all of these. I want you to understand how to use and read a karyotype. So a karyotype is a pretty ancient technique now. But it was very fundamental for understanding how DNA is organized. It was very fundamental for understanding how DNA was transmitted, and it was very fundamental for understanding some of the earliest diseases that had a genetic basis. So make sure you understand what a karyotype is and how to be able to how it is performed, and then finally how to read the karyotype to gain information about the organism or individual in question. You have to know what happens during all of the various stages of the cell cycle, which is not the same as the stages of mitosis and meiosis. Sketch the behavior of chromosomes in the various stages of mitosis and meiosis, and also recognizing in other people's sketches, which stage is being represented. So how do you distinguish between chromosomes that are isolated from mitosis? How do you distinguish between chromosomes that are isolated from meiosis? And then how you figure out which stage that it actually represents. So meiosis is very important for generating genetic variation. We don't quite understand why, but it seems to be a pretty universal truth that when animals create gametes to form offspring, there seems to be an evolutionary need for them to be distinct from one another. So this is a big part for why brothers and sisters are so different from each other, even though they share the same parents. And a large part of that variation comes from the fact that meiosis creates genetic variability among the gamete cells that are produced in this process. And this is distinguished with mitosis. So mitosis results in identical cells that have the same DNA content. Relate how the structure and activity of chromosomes determines the inheritance of genetic information, and then finally describe how defects in meiosis lead to issues in public health. So what happens if meiosis goes wrong? What are the medical consequences? Some of the examples of that will be covered at the end of the chapter. So some key background from previous classes. DNA carries genetic instructions for the development, function, growth, and reproduction for all known organisms and many viruses. Many viruses do use DNA as their genetic material. Some, for example, coronaviruses use RNA as their genetic material. But it's important to know that the reason that a species looks like a species, a cat looks like a cat, dog looks like a dog, is because they carry DNA that contains the instructions to build their body plan, etc., in a species-appropriate manner. So a DNA molecule is composed of two polynucleotide strands that coil around each other to form a double helix. So a nucleotide is a basic subunit of a DNA strand, and there's 4 different types, A, C, G, or T, and these are covalently bonded together in succession to create a single DNA strand. So a typical DNA strand is composed of millions of nucleotides arranged in a linear fashion. A is bonded to C, C is covalently bonded to G, and so forth and so forth. And these DNA strands can be amazingly long, so they can be hundreds of millions to billions of these nucleotides all covalently bonded together. So if you stretch these DNA strands out, you'd be talking on the order of centimeters to even meters long of covalently bonded nucleotides. So these are crazy long polymers. A DNA molecule is composed of two strands that coil around each other to form a double helix, and one strand is complementary to the other strand. There are also two main types of cells. So prokaryotic cells, these are bacteria and archaea cells, and these are primarily haploid, and that means that there's typically only a single copy of each genome within the cell, and DNA is organized into a single circular chromosome. So these strands of DNA, they actually come back, the ends are actually covalently bonded on each other to create a single circular chromosome. Of course, there are exceptions. There are some bacteria, species of Vibrio, for example, that have multiple circular chromosomes, and of course, many also can carry plasmids. These are very small DNA molecules, but these are very important for things like antibiotic resistance and then biotechnology used in the laboratory. This chapter is going to be covering eukaryotic types of cells, so protists, plants, fungi, animals, and these are primarily diploid. And so DNA in these cells is not organized into a single strand, but rather into multiple linear chromosomes. The ends are not bonded back together, so there's free ends of a chromosome that kind of end. These end in telomeres. And what we are interested in understanding, this chapter is primarily interested in the process of transmission of genetic material from these eukaryotic cells. So mitosis and meiosis. So meiosis is the process that leads to the production of gametes. So for two animals to have an offspring, you need to have a fusion of both a sperm and an egg cell, and each of those are haploid cells that are produced by an organism. Mitosis is involved in the production of two cells, and that's because most eukaryotic organisms are multicellular, and so the goal of mitosis is to produce exact copies of DNA within all of the cells that make up an organism. So we're going to break up this material into three major parts. So my goal is to create smaller lectures that you can digest in kind of 15-minute increments. So for this chapter, there's going to be three main parts that we cover. Part 1 will be the basics of DNA organization within the cell. Part 2 will be the details of what happens during mitosis and meiosis, the phases, the chromosomes, what they look like. And then finally, Part 3 will be the health consequences caused by defects in meiosis.